Hi, everyone. This is Ruochi Zhang from Gemma's lab. Today, I'm going to present our recent work called Multiscale and Integrative Single-Cell Hi-C Analysis. Advances in high-throughput mapping methods such as Hi-C has enabled genome-wide characterization of 3D genome features. However, the chromatin conformation actually varies across the cell population in complex tissues and also during biological processes. Such variability would be obscured when studying only the bulk data. To capture this variability, single-cell Hi-C technologies have been developed and improved through the past few years, and these technologies enabled delineation of 3D genome features in single cells, ranging from a few cells to thousands of cells. And more recently, there are new technologies that enabled joint profiling of chromatin conformation and DNA methylation in single cells on complex tissues. These exciting new technologies create new opportunities to study the variability of 3D genome structures and its functional implication. However, meanwhile, it also brings new challenges for computational methods. In particular, as compared to other single-cell modalities, single-cell Hi-C is extremely high-dimensional, sparse and noisy, and the dependency of different entries in the content maps brings extra challenges as well. And extra efforts are needed to better utilize the coacid signals such as DNA methylation. To address those issues, an end-to-end single-cell Hi-C analysis framework is needed. In particular, it should be able to generate embeddings for the single-cell Hi-C data. It should be able to impute the sparse content maps and then extract multi-scale 3D genome features from it. Potentially, it can also integrate with other single-cell modalities for joint analysis with 3D genome features. To fulfill those goals, we propose to model a single-cell Hi-C dataset as a hypergraph. So first, what is a hypergraph? I think we are all familiar with the concept of graphs where there are nodes and edges. Here in a hypergraph, we would have nodes and hyperedges, and each node can have its attributes, and each hyperedge can connect two or more than two nodes. A common example of a hypergraph would be the co-authorship network, where a publication would connect all its authors, and oftentimes there are more than two authors in a publication. So here we would model the single-cell Hi-C dataset as a hypergraph consisting of two types of nodes: the cell nodes, which corresponds to each single cell, and the bin nodes, which corresponds to each fixed size genomic bin. And every non-zero entry in a contact matrix would be modeled as a hyperedge connecting the corresponding one-cell node and two bin nodes. And when coacid signals are present, we would model them as the node attributes. So after constructing the hypergraph, we would apply a hypergraph representation learning framework to extract patterns from this hypergraph. And by extracting patterns, I mean this representation learning framework would fulfill these three goals. The one is to generate embeddings for the nodes in the hypergraph. The second is to predict the missing hyperedges in the hypergraph. And last but not least, it will predict the node attributes based on the embeddings of the nodes. And because this hypergraph is constructed from single-cell Hi-C following the procedure that I just described, you can see that these three functions of this representation learning framework is actually equivalent to the analysis goal I proposed for single-cell Hi-C, which is generate embeddings for the cells, impute the sparse content maps, and the joint modeling of coacid signals. So based on this setting, we developed this new method called Higashi. The preprint of our method is available on bioRxiv, and this method has been accepted and will be published soon. The overview of Higashi is shown in this slide. First, we would construct the hypergraph from the single-cell Hi-C dataset. After that, the hypergraph representation learning framework would generate embeddings for the cells and also impute the sparse content maps. After imputation, we can use these imputed content maps to characterize multiscale genome structures such as compartments and tags. Studying the single-cell Hi-C data using hypergraph representation learning methods would have the following advantages. First is that it integrates embeddings and data imputation for single-cell Hi-C in the same formalism. Second, it has the flexibility to analyze coacid single-cell Hi-C dataset. Last, it can incorporate the latent correlations between cells to enhance overall imputation. And by latent correlation, I mean, as compared to existing methods which would model each contact map as individual graphs, here these different cells are actually modeled in the same hypergraph, and thus the information can actually flow across different cells. And these shared information can be utilized during imputation. I will come back to this point later in the presentation to show how this latent correlation is utilized to aid the imputation. We first evaluated the embeddings generated by Higashi on several public single-cell Hi-C datasets with non-label information and made comparisons with several existing methods. The slide here shows the results we have on this SNM3C6 dataset from Joachim's lab. To evaluate the performance, we use two types of settings. One is the supervised setting, where we train a simple classifier on a small portion of the cells and predict the cell type for the rest of the cells and quantify the performance with micro and macro F1 scores. The second setting is the unsupervised setting, where we apply k-means clustering on the embeddings, and we calculate the clustering label versus the ground truth cell type with the adjusted rand index score. We can see that without the joint modeling, by just using the single-cell Hi-C, Higashi can already outperform existing baseline method here for identifying cell types. We then tested the joint modeling scheme of Higashi, and as a reminder, this joint modeling is fulfilled by training the hypergraph representation learning framework with an auxiliary task of using the embedding to approximate the methylation profiles. And we found that by incorporating the coacid signals, the performance can be further improved, and the UMAP visualization here further confirms that. As you can see, the embedding of the joint modeling in the UMAP visualization here, the cells are clustered based on the cell types very clearly. Next, we evaluate the imputation produced by Higashi. Before showing the results, I'll introduce one of the key design of Higashi. And as mentioned before, one advantage of the hypergraph setting is that the imputation can actually be coordinated across different cells. However, one concern is that what if the information sharing scheme goes too far? So we designed this mechanism to control how much information is shared. Specifically, we now have this hyperparameter k, and for each cell, during imputation, its k-nearest neighbors in the embedding space would involve in the imputation. And these k-nearest neighbors in the embedding space are likely to be cells that have very similar genome structures to this cell. And they would make contribution by using the weighted average of their content maps to construct this graph to be used as part of input to a graph convolutional network, which learns the embeddings for the genomic bin nodes. And in this presentation, all results are generated by setting k equal to 4. We evaluated the performance of Higashi for imputing content maps through a realistic simulation test. We would simulate single-cell Hi-C from this multiplex 3D genome imaging data from the Spin2 et al. Science paper. To do that, we would first transform the 3D coordinates of genomic loci from each chromosome in this imaging data to pairwise distance maps, which are referred to as ground truths and also visualized here. We will then randomly sample reads in proportion to the inverse of distance to get the sparse contact maps, which is also visualized here and marked as raw. We then apply different imputation method on the sparse contact map and made comparisons between the imputed maps and the ground truth map, and the similarity of them would be used as the imputation accuracy. Here, we mainly compare our performance to the state-of-the-art single-cell Hi-C imputation method as the high cluster, which is also developed in Joe Acker's lab. To show that the information sharing scheme is actually useful, we include the imputation results from Higashi with k equal to 0 and 4. Here, each row would correspond to one simulation sequencing depth, and each column would correspond to an imputation method. And as shown in the slide here, without using neighboring information, Higashi can already outperform the baseline methods. And by incorporating the neighboring information, the imputation accuracy is further improved, showing clearer boundaries in the contact maps as well. To show why using neighboring information can be helpful, we also visualized the cells that make contributions to the imputation of this cell and the further risk cell in the embedding space. And as you can see, the cells that help with the imputation of this cell would actually look very similar to the ground truth, while the further risk cells do not. And we run a similar simulation experiment on this new imaging data from Sweat or Cell paper and reach similar conclusions. After showing the embeddings and imputation of Higashi is actually effective, we then try to demonstrate that the imputation of Higashi could enable a detailed characterization of genome structures. We first try to call TED-like domain boundaries on this WTC11 single-cell Hi-C dataset from Binren's lab. We use the installation score-based method where the local minima of the installation score are likely to be the domain boundaries. The figure on the upper side here show the contact maps before and after imputation, and the figures on the bottom part of this slide show the heat map of single-cell installation scores both before and after imputation, and each row would be a single cell, and each column would be a genomic region. And the color red would correspond to lower installation score and thus higher probability of being a domain boundary. So before imputation, we can see that the contact maps are super sparse, and as a result, the calculated single-cell installation score is really noisy, which makes it extremely hard to robustically call domain boundaries at single-cell resolution or study the variability of domain boundaries. However, after imputation, we can see clearer domain boundary pattern in the imputed contact map, and also the installation score is much smoother. And based on these single-cell installation scores, we observe different types of variability. One is more rapid change, which we refer to as on-off switch. As you can see, for this type of variability, the boundary would disappear in a proportion of the cells. The second variability is a more gradual change, which you can, as the TED boundaries are sliding along the genome. So after observing the existence of the domain boundary variability, we then analyzed if such variability would correlate with other signals. So first, based on the occurrence frequency, we grouped the single-cell domain boundaries into 3 groups, group 1, 2, and 3, where group 3 are boundaries that appears in a large fraction of the cells, and group 1 are boundaries that appear only in a small fraction of the cells. And the scatter plot on the left-hand side here show the installation of single-cell domain boundaries and its associated occurrence frequency across the population. And each dot here would correspond to a single-cell domain boundary. We observe a negative correlation between the occurrence frequency and its associated single-cell installation score, which showing that the TED boundary with higher occurrence frequency would usually have lower single-cell installation scores. We also analyzed the single-cell binding events of these domain boundaries by using the CTCF peaks called from CTCF triad of WTC11, and we found that the domain boundaries with higher occurrence frequencies are oftentimes associated with more CTCF peaks near the anchor region and higher average CTCF peak intensity. And all these suggest that the domain boundaries that occur more often in the cell population also tend to be more stable when it appears in a single cell. Besides the domain boundaries, we also analyze the variability of single-cell AB compartment scores. We calculate the single-cell AB compartment scores, which is referred to as PC1 values for the WTC11 single-cell Hi-C dataset. The results on chromosome 21 are visualized on the right-hand side here. In this heat map, each row is a single cell and each column corresponds to a genomic region. We can see that the single-cell PC1s are overall consistent with the PC1 calculated on pooled single-cell Hi-C as well as the bulk Hi-C. Meanwhile, we can still observe the variability of single-cell AB compartment scores across the cell population. We then ask if this variability would correlate with important biological functions such as transcription. We quantify the variability of AB compartment as the standard deviation of the PC1 score, and we quantify the transcription activity variability as the coefficient of variation of the single-cell RNA-seq data. We first classify the genomic regions into regions with more stable AB compartment and regions with more variable AB compartment. And we can see that regions with more variable AB compartment would have significantly higher transcription activity variability as well. And we did the analysis from the other direction at a final resolution, where we classify genes into genes with more variable transcription activity and genes with more stable transcription activity. And we can see there is a local peak of the PC1 standard deviation for the genes with more variable transcription activity. And this analysis showed that there are correlation between the variability of genome structures and transcription activity at both global and local scale. After showcasing Higashi's ability to impute the contact maps and characterize genome structures on the cell line data, we then applied Higashi on a more complicated and challenging dataset, which is the SNM3-seq dataset mentioned in the previous slides. It consists about 4,000 cells in the human prefrontal cortex. One of the major challenges on this dataset is whether can we use chromatin conformation only to separate neuron subtypes. And surprisingly, we found that by applying Higashi on this dataset, the embeddings can already separate neuron subtypes pretty well. Then, based on the imputation of Higashi, we calculate genome-wide single-cell insulation scores as well as the single-cell AB compartment values. We found that the UMAP visualization of these 3D genome structure features can already separate the major cell types. In particular, we found that the single-cell AB compartment values can separate the inhibitory and excitatory neurons, while the single-cell insulation score has the power to further separate the neuron subtypes. So the next question would be, what type of structure at which regions contribute to the separation of these cell types in the UMAP visualization? As a start point, we analyzed if the single-cell AB compartment value near the marker genes show the corresponding cell type-specific patterns. We first identified top 200 marker genes based on the single-cell RNA-seq data for each cell type. Then, for each single cell, we calculate the average single-cell AB compartment values across these 200 marker genes. The heat map here visualizes the result where each row corresponds to a set of marker genes and each column corresponds to a single cell. We observe a positive correlation between the marker gene for a cell type and the cell type specific single-cell AB compartments. For instance, the astrocytes here would have the highest average single-cell AB values around the marker genes for astrocytes. The same applies for other major neuron and non-neuron cell types. We then took a closer look at one of the non-marker gene GAD1 for the inhibitory neuron subtypes by studying the TED-like domain boundaries surrounding the transcription start site of it. The figure on the right part of this slide shows the single cell isolation score of a 4 million base region surrounding this gene, where each row corresponds to a single cell and each column corresponds to a region. And similar as before, red color corresponds to lower isolation score and thus higher probability to be a domain boundary. The figure in the middle of the slide is also the single cell isolation, but we average the cells based on the cell type they are from. And we observe that the single cell isolation score of this region in inhibitory neuron subtypes reflect a much stronger TET-like domain boundaries, which is further manifested by comparison to the contact maps and methylation profiles. And this provides new insights into the study of marker gene regulation of these inhibitory neuron subtypes and the connection between the 3D genome structure and function. We next asked whether the genes near the cell type specific TET-like domain boundaries identified by Higashi would have distinct functional roles. We found that genes close to the ODC specific TET-like domain boundaries are strongly enriched with synapse related GO terms. We further investigated this gene THBS2, which appears in 4 of the top 5 GO term categories we identified. And this gene is known to be highly expressed in ODC and is key to the regulation of synaptic functions. The visualization of the single cell isolation scores of this region show that there are ODC-specific domain structures in a proportion of the ODC cells near this region, which can also be confirmed by the contact maps. And this demonstrates the distinct ability and advantages of Higashi to effectively identify cell types and cell type-specific 3D genome features in complex tissues. In summary, we developed this method called Higashi as an end-to-end solution for single-cell scHi-C analysis. Besides the main Higashi algorithm, we also developed this visualization tool to facilitate the single-cell scHi-C analysis. One of the key innovations of this visualization tool is that it allows synchronized navigation of the UMAP visualization of the embeddings and the contact maps. For the next steps, we plan to first improve the imputation ability of Higashi, especially for the long-range contacts. Moreover, the hypergraph setting of Higashi allows more than 3 nodes in a hyperedge and thus can be extended to study the single-cell multi-way chromatin interactions, such as the single-cell sprite data. We also plan to integrate Higashi with other single-cell modalities as well as analysis results. The preprint of our work is available on bioRxiv. The code for both the Higashi main program and the visualization tool is available on GitHub, where we have a detailed documentation. Last, I would like to thank my advisor, the co-author of this work, and the other members of our lab. I would also like to thank members in the 4DM group, especially members in the 4DM UM1 Center and the NoFIC AICS working group, where this project receives a lot of useful feedbacks. Thank you.